Um, I am Carlos Arteaga from UT Southwestern Simmons Cancer Center and co-chair for AACR for this meeting. And I've been asked to moderate the spotlight discussion session titled Genomic and Genetic Analysis on Metastases. The format we will follow is the following. Dr. Christina Curtis from Stanford University will do a virtual discussion of posters 2, 3, 4, and 5, followed by your questions that should be submitted via the app to Drs. Jane, Bravanel, Louis, and Chibert. After that, after we handle those questions, we'll go to the next discussant, who is Dr. Daniel Stover at The Ohio State University Comprehensive Cancer Center, who will discuss posters 6, 7, and 8, after which we will receive individual questions to Dr. Kong, Dr. Son, and Dr. Garatana. So let's start then with the discussion by Dr. Christina Curtis from Stanford.
My name is Christina Curtis, and it is my pleasure to chair this spotlight session on genomic and genetic analyses of metastases. In this session, we'll focus on advances in understanding the somatic genomic and immune landscape of metastatic breast cancer, while also touching on germline genetic features and the use of self-recirculating tumor DNA to monitor disease progression. I will cover the first 4 abstracts in this session, and Dr. Daniel Stover from the Ohio State Comprehensive Cancer Center will cover the remaining 3. And these are my disclosures. So the past few years have really shown us tremendous insights into the genomes of metastatic breast cancer. And in particular, a seminal study from the Memorial Sloan Kettering group profiled over 1,500 hormone receptor positive tumors with their impact panel, for which detailed clinical information and treatment outcomes were available. Now, over 700 of these patients had been previously exposed to hormone therapy, and this study revealed a number of alterations in the MAP kinase pathway, as well as ERB2 activating mutations, NF1 loss of function mutations, and alterations in estrogen receptor transcriptional regulators. Subsequently, using whole genome sequencing, and in some cases augmented by RNA sequencing, others have sought to further characterize the landscape of metastatic disease. And again, often comparisons have been made to unpaired primary tumors due to the challenges in obtaining paired primaries in mets in many instances. Now, these studies have started to enable insights into the impact of therapy on cancer genomes. In this study from Pitch et al., they demonstrated a variety of mutational processes found in metastatic lesions, and in particular, they found a capecitabine signature, which really was characterized by the enrichment of T to G transversions. And in work from my own group, in a comparison of exome sequencing data from paired primaries and distant metastases, we found that there was an enrichment for private clonal driver events in the metastases amongst patients who were treated. These metastasis private drivers tended to be enriched for chromatin modifying genes, consistent with their role in mediating epigenetic plasticity during disease progression and in the context of therapy. Now, fast forward to this year, and the Aurora project has characterized the genome and transcriptome of paired primaries and matched metastases in the context of clinical care. They had identified subtype switching amongst intrinsic subgroups in roughly 37% of cases, predominantly amongst luminal Bs. And they also found that more than half of all patients had SCAT tier 1 or 2 alterations, speaking to the importance of a metastatic biopsy and the potential actionability of these alterations. Now, while the studies to date that I've described have leveraged bulk profiling, it is also the case that single cell and spatial approaches are coming of age. And in this tour de force study from Alex Swarbrick's group, they profiled the landscape of 26 primary tumors over 130,000 cells and can start to chart the cellular states as well as composition amongst subtypes. And now these tools are also being used to study metastatic lesions, as we'll hear about in some of our posters today. So the first poster is abstract 1134 from Jane and colleagues, entitled "The Metastatic Breast Cancer Project, Expanding the Clinical, Genomic and Transcriptomic Landscape of Metastatic Breast Cancer Through Patient-Partnered Research." And really the goal of this is to create a publicly available data set linked to clinical genomics. And their approach has been to leverage robust patient engagement through social media as well as advocacy. And as you can see, they've consented over 3,000 patients from 1,700 institutions between 2015 and 2020. And really these rich clinical patient-reported data is accompanied by comprehensive genomics via whole exome and RNA sequencing. So with the patient-reported data, one can begin to drill into the time between primary and metastatic diagnosis, and we see that really there's most mass over here in the 0 to 4 month time period, so relatively short time span. And we can also use these data to look at the treatment course across a variety of agents for each of these patients. And what is quite impressive in my view is that there was a very high average concordance across different patient-reported data with the information abstracted from medical records. Now with the RNA-seq data in hand, one can start to survey for in-frame fusions, particularly those with functional events. And over 81% of tumors had at least one fusion. Roughly 20% of those were found in OncoKB, and some examples of breast cancer-associated genes are seen here. But this really also highlights the fact that these alterations tend not to be highly recurrent. We see a lot of different genes represented. And with the RNA-seq data, the authors also investigated PAM50 subtype switching in a cohort of 35 patients with paired primaries and mets, and they found that 42% exhibited switching, again, predominantly amongst luminals, and this was similar to what was seen in the Aurora project. Now, one can also survey the actionable mutations that are present, and we see that almost 50% of patients had a PI3 kinase mutation, just under 40 had an ESR1 mutation, and there were other alterations as well, with varying degrees of actionability, levels of evidence. Using the germline data, one could also survey for likely pathogenic germline variants that occur in concert with somatic alterations, as shown here, an NF1 ATR at the top, followed by SMARCA1 and BRCA2, and roughly 11% of patients were found to have such events. Now with these data, one can also survey the landscape of metastatic disease, and many recurrent cancer genes were found that have been previously reported to be enriched in metastases, particularly in treated tumors, namely ESR1, but also P53, PIK3CA, and others. And importantly, these landscapes can be compared with patients with de novo and/or treatment-naive disease, and I think there's a lot of interest in doing that. Now, for a subset of patients, multiple biopsies, both tissue and blood-based, have been obtained. And as we can see for this case study here, 2 distinct ESR1 hotspot mutations were observed at time point 2 and time point 4. And additionally, via RNA sequencing and activating an in-frame ESR1 fusion event was detected. And so really what this speaks to is the observation that these repeat biopsies can reveal convergent evolution and mechanisms of resistance. We look forward to further studies of this in longitudinal samples. So in summary, we're really seeing an expansion of clinical genomic data sets for metastatic breast cancer, and we'll hear more about those. The Metastatic Breast Cancer Project is leading the way in this and is really a rich resource to the community. They make regular data releases available on CBio portal as well as dbGaP. And this makes it increasingly feasible to study unique clinical cohorts such as young patients, rare subtypes, as well as biomarkers of response and resistance. And so my questions to the authors are, can you begin to start to survey associations with exceptional responses, as has been reported by patients? Can you also start to investigate the landscape of de novo stage 4 disease relative to treatment-naive tumors at both the genomic and ultimately at the immune level? And can you comment on the attrition rate for the different analytes that are used, given that these are from clinical samples, and there must be attrition at both the sample and data QC level. So moving on to abstract 1170, entitled Spatial-molecular dissection of breast cancer metastatic environment from Abravanel and colleagues. This study sought to establish a prospective metastatic breast cancer research biopsy collection protocol to enable both single cell and spatial profiling, and really to facilitate comparison of characteristics across different dissociative and spatial technologies. Now, the authors established a robust research core biopsy protocol, really seeking to minimize time from sample acquisition to processing. And with this approach in hand, they were then able to survey different types of approaches, both single-cell RNA-seq and single nuclei RNA-seq, as well as spatial approaches with varying degrees of resolution, and those that are both RNA and or protein-based. And so this provides a really rich opportunity to compare across these methods. Now, in the first instance, 67 metastatic breast cancer biopsies were taken that were split for either single nuclei RNA-seq or single cell RNA sequencing. And you can see that the number of cells was greater for the single nuclei protocol versus the single cell, as was the number of median genes per cell. But with both of these approaches, we can start to parse out differences in the composition and prevalence of certain cell types, such as lymphoid, malignant, myeloid, or stromal, across individual patient biopsies, and there are indeed differences. Notably, one observes a greater degree of hepatocytes in the single nuclei RNA sequencing, as well as the higher proportion of malignant and stromal cells and stromal content versus the single cell protocol. And in contrast, a higher proportion of lymphoid and myeloid populations were seen in the single cell protocol. And the authors will continue to investigate these differences. Importantly, these were separate biopsies, so they are not a one-to-one comparison. And with these data, one can also start to explore inter and intra-heterogeneity in receptor expression within the clinical subgroups, and this is shown here in these violin plots for the single nuclei RNA data. In particular, what one can see is that there can be bimodal distributions for individual markers within a single tumor biopsy, suggesting that there are populations with either high or low expression of either ESR1, PR, or HER2. And I think that's quite intriguing. There is, of course, variability across the population in these different markers, but this really gives us a lens into heterogeneity within these clinical subgroupings. Now, with the spatial data, one can start to perform cross-platform comparisons, for example, using SlideSeq, MERFISH, and Codex. And we can see that different cell types are in fact represented here. You can zoom in and start to see the enrichment of endothelial vascular amongst alongside fibroblast populations and the like. And in fact, with these data and combining them with the dissociative single cell RNA-seq, one can start to measure the relative co-occurrence or neighborhoods of different cells. So looking at the distance between different cell types and endothelial cells or fibroblasts. And so ongoing efforts will continue to leverage new analytic methods to interpret these data, but this provides a promising area of new research. So in summary, really single cell and spatial profiling has come of age. It is feasible not only in primary breast tumors, as I showed you in the beginning, but also in metastatic lesions. And these types of approaches can reveal cell types, states, topography, and their interactions. It's hoped that this kind of cross-platform analysis will inform the best use cases for certain technologies. And yet, I would also point out that it's still early days, that other spatial methods are still on the horizon, and really that new analytic methods will be critical for parsing and integrating these rich data. And so my questions for the authors are really, where do you envision the greatest bang for buck? These are expensive and complicated technologies. Could we get away with just building an atlas and then integrating with bulk data from there? And what do you think about the extension to serial biopsies really as a means to study the impact of therapy? Of course, that requires that multiple samples pass the QC, perhaps across different platforms. And so leading to my last question, which is really, can you comment on the success rate across these different platforms and the ease of use? So now on to abstract 451, entitled The Mutational Landscape and Immune Infiltration of Breast Cancer Metastases to Gynecologic and Other Organs. This study was led by Dr. Louie and colleagues, and their goal was really to investigate the association between mutations, subtypes, and sites of metastasis. They employed the CARIS dataset of over 12,000 breast cancer patients. Roughly 5,000 of those were primary tumors. The vast majority were METs, as you can see over here. And these data were profiled, these samples were profiled using either a 592 gene panel or whole exome sequencing. And this was complemented by RNA sequencing, as well as PD-L1 immunohistochemistry. And really their goal was to focus on cancer relevant genes and their prevalence, as well as tumor mutational burden. So what's apparent looking at the landscape of these cases is that if we focus in here on brain metastasis, it's apparent that triple negative really dominates with 38%, followed by roughly 28% for HER2 positive disease, as well as hormone receptor positive HER2 negative disease. It's also apparent that METs to gynecologic organs, as shown over here, really were dominated by hormone receptor positive tumors. And that's somewhat intriguing given that these patients were, with gyn METs, tended to be younger, on average age 53 versus 16. And so one can now survey the differences in both mutational burden and other features. And we see a decrease in 53 mutations in METs, as well as an increase in PIK3CA, ESR1, lower PD-L1, and also increased tumor mutational burden. And so those are sort of features that we might expect to see, not terribly unsurprising. But one can also start to chart other broad stroke pathways according to metastatic site, looking at brain, liver, and beyond. And of note, it was observed that in gynecologic metastases, they tended to be enriched for patients with lobular histology. There was a reduction in PD-L1 levels, reduction in p53 and homologous recombination, and a concomitant increase in PI3 kinase pathway, Wnt signaling, and luminal A subtypes. And so drilling into the mutational landscape further, across different metastatic sites for a set of cancer-associated genes, we really start to see this enrichment of mutations, including BRAF, MAP3K1, FOXA1, CDH1, PI3 kinase, in gynecologic organs. The CDH1 alterations were shared as well in GI organs. And also using the RNA sequencing data, it was apparent that there was an increase in the M2-M1 macrophage ratio relative to other organs and specifically breast primary tumors. And this was based on quanti-seq deconvolution. So in summary, site-specific differences in mutations and immune composition are apparent. There's an enrichment for specific mutations in gyn-METs, and they also harbor an immune suppressive landscape. Now, it is currently unclear whether these features are shared by primaries given the lack of a paired sample, but it's possible that mutational clonality may shed some light on this in terms of the relative selection and timing of these alterations. And in addition to that, the status of CDH1 mutations as is particularly relevant here in the context of high-frequency germline events. So my question for the authors is, what is your speculation around these patterns and particularly the different immune suppressive microenvironment? Is this just a function of the architecture and features of those organs, or might specific alterations also contribute and modulate the microenvironment? And additionally, given the enrichment for actionable mutations, can you comment on the implications for repeat biopsy and moreover, whether one would anticipate any heterogeneity of those sites? So lastly, moving on to abstract 323 from Chibber and colleagues, entitled Characterization of the Immune Landscape of Primary and Metastatic Breast Cancer in a Real-World Cohort. So the goal of this study was to survey the immune microenvironment of primary and metastatic breast tumors in a clinicogenomic real-world dataset. This was from Tempus. The dataset was comprised of RNA sequencing from 666 biopsies from various sites, and you can see these here. I'll point out that the majority of cases had stage 4 disease, a subset had stage 1, and that's really the majority of 10%, but the majority of stage 1 through 3 were stage 1. And also to say that there's a number of metastatic biopsies from other sites that are not annotated. And the authors then used these RNA sequencing data to infer immune expression signatures and composition using estimate-based deconvolution. So in the first instance, looking at primary tumors, we can see that TNBC has a higher immune infiltration signature than hormone receptor positive disease, as we would expect. And in stage 4 disease, these patterns were comparable. Of note, in triple negative breast cancer, there was a decrease amongst patients with stage 4 disease, presumably those that were de novo metastatic. And this is no doubt intriguing, but I would point out again that these patients in the stage 1 through 3 were predominantly stage 1. And then there was really no difference in the hormone receptor positive group. And so this is an interesting observation. Now, using, again, the expression data, they sought to investigate macrophage scores across stage, early stage breast cancer, stage 4, and different metastatic sites. And here in the hormone receptor positive HER2 negative subgroup, we see that there is an elevation of these macrophage profiles in stage 4 and liver disease. And similar profiles were seen for M2 macrophages as well. Now, they also sought to investigate patterns of immune infiltration across metastatic site. So starting with the brain, it's apparent that patients that had a brain metastasis and where the brain was profiled, that these patterns are distinct from other metastatic sites as well as from primary breast tumors. This was similarly the case for lung biopsies, and yet liver biopsies tended to be more similar to patients that had liver metastases, but for whom a different site was biopsied. And so this is an intriguing observation. I would say that they observed similar patterns for specific immune signatures such as CD8 positive cells. And this is really in line with prior reports that CD8 T cell density is lower among patients with liver mets, irrespective of the site of biopsy. They also investigated the expression level of key immunotherapy targets, and they found that CTLA-4 and PD-1 tend to have higher expression in primary tumors as compared to metastases. And this has implications, perhaps, for how we think about selecting patients for treatment on the basis of immune markers. So in summary, the authors have shown really that the immune landscape varies across stage and metastatic site. We see that brain and liver mets have lower immune signatures, and that includes those signatures that are associated with response to immunotherapy relative to primary breast tumors. And higher M2 macrophages in brain mets is consistent with their immune suppressive TME and some of the challenges faced in treating patients with IO. And so my questions for the authors are, can you speculate on the lower immune scores seen in TNBC primary tumors? Were similar patterns observed in stage 3 disease? Also, why might the immune composition of liver mets be more similar to other sites amongst patients with liver disease? And, again, does this play into the possibility of reseeding or colonization that would predominantly be observed for mutation-based profiles? But I think this is an intriguing observation that the community may need to be thinking about. And then finally, can you speak to the implications for treatment selection and really the next steps in corroborating these findings? So I want to thank the poster authors for really sharing their exciting work, for sharing with me their slides in advance. I want to thank Drs. Arteaga and Stover for their participation in this session, as well as the patients and their families who enable and inspire this research. Thank you.
Thank you, Dr. Curtis. Please don't leave, stay there. Any questions for any of the posters, starting with poster number, the second poster, PD602, the Metastatic Breast Cancer Project, expanding the clinical genomic and transcriptomic landscape of metastatic breast cancer through patient-partnered research. I don't see any questions in the app, but maybe I see 2 microphones in the back. Maybe if anybody has a question, please speak up.
Sure, and I can kick it off while we're waiting to hear from the audience. So to Dr. Jain, I think this was really a tremendous, it's a tour de force, this project. I think it'll be an immense resource for the community. I was really curious about these exceptional responses, and I wondered if you could speak to what you might be able to already learn from the 31% of patients who have reported that so far.
Thank you, Dr. Curtis, for your question, and thank you for letting me participate virtually in this meeting. To your question, we did start comparing the patient-reported extraordinary responses to the already the clinical data or the treatment data that we have from about 250 patients, and see if the responses actually correlate in the patient-reported data versus the medical record data. We have seen several cases where the correspondence actually matches in different drug categories. For example, we have seen a patient that responded extraordinarily to TDM1, for example, and we could find certain genomic correlates that were associated with the response to TDM1, and which was basically reported both in the patient-reported data as well as validated from the clinical records that we abstracted here. So we are seeing cases like that, but we haven't certainly done a full sort of deep dive into all the patients that that marks themselves, that we have basically identified through the patient-reported data.
Thank you. Thank you. Okay, so maybe we can go to the next questions for PD603, spatial-molecular dissection of the breast cancer metastatic microenvironment. You have any questions, please send it through the app or come to the microphone, or maybe Christina, I think you had a question about the concordance of the platforms or something like that.
That's right. That's right. Yeah, I'd love to follow up on this. I think, I mean, it's again, huge amount of effort to profile with these different platforms. They're also expensive technologies. You need a lot of in-house know-how to man them. So I was just wondering if you could speak to where you see this really rolling out in the field, and whether there's the possibility of leveraging atlases like this to integrate with the more common bulk sequencing data that is out there.
And this is for Dr. Abravanel, correct?
Yes.
Correct.
Yeah.
Thank you so much for the question and the wonderful discussion. I absolutely agree that it takes a lot of effort to profile with all these different methods as we've set out to do here. And we really did start off doing this as a resource to develop the techniques and the pipelines for the community to be able to understand and compare how to use these different techniques. Of course, it turns out that there is no one that substitutes for all the rest. They each have their own role with different logistics, different cost and technical know-how requirements, and also different outputs. And so it really depends probably on the question that one is trying to ask as to how you should approach, which you should choose and how you should go about that, what samples you have and what questions you're asking. In terms of trying to use these kinds of data to leverage more common data, we're very excited about this possibility. Because as you know, in our group, we also have large amounts of exome and RNA-seq data from bulk populations. And so I think this is definitely an important direction for the field to go in, is going back and forth between these data sets. And so once we're able to sort of put this resource out there to interrogate it, not just by adding additional single cell or spatial data, but by using it to either deconvolute or help better understand some of the bulk data that we're looking at. And I think one of the other questions you asked about were serial samples. And so I'll just take 2 seconds and say we're very interested in this as well. In the data set that I've presented so far, there aren't any serial samples, but we have a number of ongoing projects, including doing just this. And so, for example, one area that we're very interested in the group is understanding mechanisms of response and resistance to CDK4/6 inhibitors. And so we have an ongoing project to do sort of a subset of these profiling techniques on biopsies immediately before and after patients go on and off CDK4/6 inhibitors. And some of those will be serial samples where we'll have the opportunity to sort of dive deeper into the areas you were describing.
Thank you. Thank you very much, Dr. Abravanel. Let's go to the next abstract, is PD604. mutational landscape and immune infiltration of breast cancer metastases to gynecologic and other organs. And we have the first author, Dr. Louis, here joining me at the podium. So, Christina, I may need a question from you, or I can ask something too.
Go ahead if you have one. I'm happy to chime in, but please go ahead.
Well, I was just going to ask Dr. Louis whether she can speculate as to what would be the mechanistic basis for this, what appears to be a mutational specificity to this, tropism to gynecological organs.
So this would be at least partially speculation. I mean, we know from other prior work that there are subtype specificities to, like, for instance, lobular cancer is much more likely to go to gynecologic and GI organs. So there's definitely some difference in the cancers that go to those organs already. As far as what genes are causing that, or if it's some subsets that of the primary tumor cells that develop it in the primary versus is this something that there's a selection factor in the metastasis? That's because I also am limited as far as not having matched samples is something that's really hard for me to answer. I think it's probably a mix of those factors would be my speculation that other studies have shown that individual metastatic sites tend to be more homogeneous than the primary tumor. So there is certainly some aspect of single clones that are able to survive, but also which one in certain studies there are there are certain clones that are selected by the organ itself that they end up metastasizing to. So I think that there's a lot more work to be done to figure out what is making certain metastases show up with these particular patterns. But I do think that it does lean towards potentially the importance of repeating biopsies, particularly if there is a particular metastatic site that is not responding to therapy. It may have different targetable mutations that are not obvious elsewhere.
Yeah. So thank you. So the neighborhood matters for the oncogene, I guess. So that's the lesson, and I think this is going to be it's going to need further work. But good luck. Thanks for the answer. Let's go to the last one, which is characterization of the immune landscape of primary versus metastatic breast cancer in a real-world data cohort. I think Dr. Chiverri is not here. He's probably on Zoom. And Christina, do you have a question for him?
Sure. Yeah, I was really intrigued by what I think was the observation that de novo triple negative breast cancers that were de novo stage 4 had quite lower immune scores than earlier stage tumors. And I recognize that those were predominantly stage 1, but I did just want to confirm that those were de novo metastatic patients. And if you could speculate about the immune microenvironment that we're seeing in these patients.
So just to clarify, Christina, you mean these are previously untreated? Is that what you're?
Well, what I was curious about is, number one, they're listed as being stage 4, but were they de novo stage 4 based on the reading of the data that I could extract from the poster? It seemed to imply that to me, but I wanted confirmation. And, then yes, just to sort of ask the question, I mean, do you think that if you were to compare with stage 3 disease, that we would actually see a major difference? Or is this because we were really comparing stage 4 versus stage 1 for triple negative?
Yeah, to the last point, as you pointed out, our early stage 1 through 3 was, primarily stage 1, and we just didn't have enough data points in the stage 3. So I can't speak to the, whether this would be a continuous trend that we would see, or whether this is because we're looking at the two extremes, which might be, what we're looking at. Although it does look like the stage 4 is kind of, with respect to immune infiltration, looking a little bit more like the metastases from other sites, which is interesting. So these could be previously treated. If I understood your first question correctly, and I may not have, these could be, the biopsies could be coming from subjects who were previously treated and have progressed to stage 4.
So you don't have that annotation, that you don't have the resolution to resolve that. Yeah, and that was part of my question is what's inducing this reduction in immune microenvironment. If it were true of de novo disease, it would be very interesting to understand that better, of course. So
it's a real world data set, and it's a very rich data set, and there's lots of things we can explore, but one challenge is kind of missing information and dealing with a little bit of the noise in the data set.
Yep.
Absolutely.
Good. Thank you. There's a question in the back.
Hi, Fraser Simmons from MD Anderson. Just a quick question. It's very interesting that triple negative, that the stage ones, I'm wondering if they might have been enriched a little for operable cancers with, sort of medullary-like characteristics, a lot of lymphoplasmacytic infiltrate. Early stage triple negative can be quite different from the more aggressive variants that are seen in the metastatic setting.
Yep.
Okay, thanks for that comment. So thank you, Dr. Chiver. We'll go now to the